Hello everyone and welcome to our webinar titled Parkinson's Disease, Unveiling the Promise of Neuromelanin Sensitive Imaging. My name is Hiba Kazmi and I will be your host and moderator for today's session. Before we start, I just wanted to go over some housekeeping points. Please ensure your audio settings are set to computer audio. We'll also be addressing any questions during the Q&A session at the end, so please do feel free to ask questions at any time using the Q&A box on your GoToWebinar panel. Finally, this webinar is being recorded and you'll receive a link to the recording after the session. Now onto our big topic of discussion, the use of MRI to measure longitudinal change in clinical trials of Parkinson's disease. In particular, research into neuromelanin MRI has been ongoing. We are now seeing its use and results in larger multi-center trials, giving us a lot to think about for its application in the future. This webinar will cover the significance of neuromelanin in Parkinson's disease and the more practical considerations required when setting up this sequence. With that, I'm very pleased to introduce our two wonderful speakers. First up, we'll have Professor Stéphane Lehéricy, who is a professor of neuroradiology and head of the Center of Neuroimaging Research at the Paris Brain Institute. He has extensive experience in the structural and functional study of the human brain, focusing on movement disorders and neurodegenerative dementia. Stéphane has significantly contributed to understanding the functional anatomy of the basal ganglia using advanced imaging techniques. Our second speaker will be Vahid Malekian, who is a biomarker scientist at IXICO, specializing in MRI data acquisition, analysis, and biomarker development. With a PhD in biomedical engineering, Vahid has worked on various projects related to MR pulse sequence development and image analysis. He collaborates with pharmaceutical companies to integrate MRI biomarkers into clinical trials. As for myself, I am a senior biomarker scientist and the imaging lead for Parkinson's disease and SPECT imaging at IXICO. I have experience in movement disorders and psychiatry, with my PhD focusing on clinical and imaging features associated with prodromal Parkinson's disease and those with late-onset depression. My focus at IXICO is on consultancy for clinical trials across TAs. Now over to our first speaker, Stéphane, and our talk, which will be Neuromelanin-Sensitive MRI, a biomarker of substantia nigra degeneration in Parkinson's disease. Over to you, Stéphane.
Thank you for the introduction. Hello, everyone, to attend this session. I'm going to talk about neuromelanin-sensitive MRI as a biomarker for substantia nigra degeneration in PD. The next. We have three parts, a brief part on the introduction to Parkinson's disease, then the neuromelanin-sensitive MRI as a biomarker, and one slide on the practical and technical challenges with neuromelanin-sensitive MRI. Next. Well, as you know, I guess most of you, Parkinson's disease is the second most common neurodegenerative diseases in the world, with the prevalence estimated to be 1.5 per 1,000. Men are more affected. The prevalence increases with age and with time. It is supposed to be plus 74% from 1990 to 2016, and the prevalence is predicted to rise over 12 million worldwide by 2040. The pathophysiological hallmark of PD is abnormal aggregation of alpha-synuclein and Lewy pathology, and also, as you know, degeneration of dopaminergic neurons in the substantia nigra pars compacta. Next, please. The clinical presentation of Parkinson's disease is, of course, the motor symptoms that are the hallmark of PD, with bradykinesia, rigidity, rest tremor, and postural instability. Next, and this is mostly due to changes and neurodegenerative changes in the substantia nigra, which is also linked to cognition. And next, in PD, we have also non-motor symptoms that sometimes very often predates the occurrence of motor symptoms, with autonomic dysfunction that are also related to medulla oblongata changes, REM sleep behavioral disorder with a locus coeruleus, pedunculopontine nucleus with postural instability, locus coeruleus and cortex with cognition. Next. Well, the imaging biomarkers of nigral degenerations include radio tracers with PET and SPECT, mostly for the vesicular monoamic transporter like VMAT2, and for the membrane dopamine transporter, and the most known is DAT-scan with euflupan. And also fluorodopa with PET for the activity of dopa decarboxylase. Next. We can also image the dopamine receptor ligands, which are postsynaptic tracers. And with MRI, next, We have at the moment 4 biomarkers that are being investigated. One is neuromelanin imaging, which is the topic of this talk. The second one is the swallow's tail sign. The third one is iron imaging with quantitative susceptibility mapping, and the last one is free water with diffusion imaging. So now we'll focus on neuromelanin imaging. Next slide, please. As you know, neuromelanin is a marker of catecholaminergic neurons in the substantia nigra, dopaminergic and locus aureus, noradrenergic. On the image, you can see neuromelanin as these dark granules in the dopaminergic substantia nigra neurons. Next. The origin of neuromelanin signal is debated. Mostly is due to paramagnetic T1 shortening effect of the complex neuromelanin and iron, but also probably to the magnetization transfer effect due to the high content of paramagnetic iron-associated water proton in the nigra. And that will be the topic of the next talk. And this is the aspect in a normal subject where you can see here the substantia nigra as this high signal intensity band on the T1 weighted image. And in PD, next, what you can see is a reduction of the signal intensity and also reduction of the volume of the substantia nigra. This is a clinical biomarker next, because as shown here on the example, on the upper row, you have 3 healthy subjects where you can clearly see the substantia nigra as the high band of signal, the high signal intensity band here on both sides. And on the lower row, you have 3 patients with Parkinson's disease, and clearly you see the reduction in volume and signal intensity. And by just radiological reading, like the radiologists can do, just with a naked eye, then we have 85% accuracy to distinguish PD patients from controls. The next slide. We'll show you also that measurements can be derived from neuromelanin sensitive imaging, and that will be also the topic of the next presentation. We can use manual segmentation, template segmentation, deep learning approaches to obtain measurements, which include the volume and the signal intensity. And with that, we can derive a number of biomarkers. So next, please. The first one is the fact that neuromelanin is a longitudinal biomarker. So neuromelanin will decrease, the signal intensity of the volume will decrease over time, like in these 2 studies here. Next, where you see the volume here corrected by the total intracranial volume from healthy controls on the left to advanced PD patient, 12 years disease duration on the right, and clearly you see a reduction in the volume over time. Next, please. With this representation, which is a group map of several subjects here. The red color means that you have more neuromelanin, and you have here healthy controls on the left, where you clearly see the shape of the substantia nigra. And then the reduction from early PD, 1.5 year disease duration, to advanced PD with a 12 years disease duration. Clearly, you see that neuromelanin MRI is a longitudinal biomarker. Next, please. It has been also shown that it's an early biomarker because it has been evidenced in patients with isolated REM sleep behavior disorder, which is considered the prodromal stage of Parkinson's disease, that you see a reduction in the signal intensity and volume here on the image on the left, and on the measurements on the right with the automated segmentation with the healthy volunteers on the left and the PD patients on the right and the isolated RBD in the middle, where you also see a reduction in the volume of the substantia nigra. Next, please. It's even present, the reduction in volume in asymptomatic mutation carriers. Here it is LRRK2 mutation carriers. That's a study conducted in collaboration with Tao Wu in Beijing, where you have here the reduction in volume of the substantia nigra using neuromelanin signal in LRRK2 asymptomatic carriers in between healthy controls and PD patients. Next, please. It correlates with striatal dopamine transporter binding. On the left, it's PE2I in the dorsal putamen. So you have a correlation between neuromelanin, that's the x-axis, and the labeling in the striatum on the y-axis. And next, this is a study conducted by our group between neuromelanin and DAT scan in the sensorimotor putamen, conducted using a voxel-based image. So here you see the signal intensity in the substantia nigra using neuromelanin imaging that correlates with that in the sensorimotor putamen. Next, please. We can also follow the volume of the substantia nigra over time. On the x-axis, you have here the disease duration. 0 means that it's the onset of the motor symptoms in PD. And next, please, we have estimated that it is possible to detect changes in the substantia nigra starting 5 to 6 years before the onset of the motor symptoms, when the signal reduction is about 23%. Signal and volume reduction is about 23%. And this has been reproduced, next please, with an independent study here by the group of Dorothea Auer in Nottingham, where she reported exactly the same disease duration for the onset of changes in the substantia nigra with neuromelanin imaging. Next, please. If we look at the changes in the different sensorimotor, associative, and limbic compartment in the striatum, so this is the DAT scan. It's a coronal image. More red or darker red means that it is more affected. That's the sensorimotor striatum, it's the putamen. Then the associative in light red, and in orange you have the limbic putamen. And we have estimated the beginning, the onset of the changes. Next, please. In the sensorimotor putamen, here in blue, starting between -10 and -20 years before the onset of the disease. And then you have the associative territory in green and the limbic territory in pink. And for neuromelanin on the right, next please, it's the same. So the changes will start here in the sensorimotor territory. That's the posterior and lateral part of the substantia nigra between 5 and 6 years before the disease onset. And this corresponds to the sensorimotor territory of the nigra. Next, please. The neuromelanin signal changes correlate with motor, cognitive, and behavioral symptoms. It's also a voxel-based analysis between the signal intensity in the nigra and the UPDRS3 on the left, so in blue, and you see that the changes are mostly found in the sensorimotor part of the nigra, the posterior and lateral part of the nigra. The cognitive changes, that's a correlation with the Matisse scale. It's mostly in the more medial and anterior part of the nigra. And for the behavioral scale, it's called the Ardouin scale with the apathy subscore and the creativity subscore. It's really more medial, close to the ventral tegmental area. So MRI changes correlate with motor, cognitive, and behavioral symptoms. Next, please. Signal changes also are influenced by the genetic risk score of patients. Here you have the Parkinsonian risk score that was obtained using 19 SNPs, according to that paper of Niles and colleagues, with a statistical model adjusted for age, sex, and the delay since diagnosis. You see that there is no correlation in healthy controls, but you do see a steeper decrease in the volume of the substantia nigra in patients with a high PRS polygenic risk score compared to patients with low polygenic risk score in green. So the high polygenic risk score is in red and the low polygenic risk score is in green. So the higher the polygenic risk score, then the steeper the decline over time. Next, please. Now, one question is the histological correlate of neuromelanin signal intensity that we detect with MRI. And for that, we have used a model that has been developed by Miquel Vila in Barcelona, where we have injected human tyrosinase in rodents. These are rats that do not produce neuromelanin in the wild type. And with the injection of the viral vector and human tyrosinase injected right above the substantia nigra pars compacta, then the animal will develop neuromelanin, and this can be seen here on the lower row, where you have an image of the dark band in the macroscopic view of the substantia nigra as compared to human on the upper row. It's also the same type of changes that you see on the hematoxylin eosin image in the middle, and on the right you have the electron microscopy. So the changes in the substantia nigra are very similar to the ones observed in humans. What we can see on the next slide is the fact that with the MRI, it's a 11.7 Tesla Bruker system with the rat model. On the right of the image, you can see an elevated signal intensity in the substantia nigra on the side where we did the injection of the viral vector plus the human tyrosinase. Next slide, please. And what we have observed here is that if we do a comparison between the ipsilateral, the same side as the injection, and the contralateral side, and we compare before the injections, that's the -1, I'm at -1 month, then you don't have any difference between the two sides. So the ratio is 0. After 1 month post-injection, you clearly see an increase in signal intensity on the injected side, and a reduction that starts after 1-2 months after the injection with a slow decrease of the signal intensity. Next, if we look at the histological changes in these animals, what we have observed is that if we quantify the intracellular neuromelanin into the nigral cells, that this is shown by the red arrows on the lower image. You can clearly see here the neuromelanin into the cells. And if we quantify neuromelanin intracellularly using optical density on the upper image, at 1 month post-injections, 2, 4, and 8 months post-injections, what we see is a steady increase, which is regular over time with no threshold. And next, if we look at the number of Th positive cells, which correspond to the dopaminergic cell bodies, what we see is that we start seeing the generation of these cells after 2 months post-injection with a reduction of the number of cells at 4 months and 8 months post-injections with a ratio of the IPSI over contralateral, which decreases below 40%. And on the lower image on the contralateral side, which is not injected, in blue you see the Th positive cells, and the lower image with the injected side, you see the neuromelanin and very few remaining dopaminergic cells. Next, please. Now we can better interpret what we have seen with the neuromelanin image with MRI. The first part of the curve is here the accumulation of neuromelanin intracellularly up to a certain threshold at 1 month, 2 months. And then what we see is that degeneration of the dopaminergic cell will take over, and we have a reduction of the number of dopaminergic cells and also in the signal intensity of neuromelanin in the substantia nigra. Next, using this knowledge, what we can do is better interpret that curve, which is the curve produced by the group of Dorothee Hauer in Nottingham, where they have examined over age here from about 3 to 5 years up to 90 years, the signal intensity with age. Clearly, you have a strong increase from childhood to adolescence up to a plateau in the middle age between 50 and 60 years, and then you have a decrease in the older age. So probably what you see is an accumulation of neuromelanin into the cells from childhood to the middle age, and then you start seeing neurodegeneration of these cells in the older age. This is in normal controls. And in patients with Parkinson's disease, it's steeper, and the degeneration is increased. Next, please. What we have done also is, this is only a single-site study. We have tried to determine whether with neuromelanin imaging, we can try to detect changes that could be used, for instance, in a therapeutic trial. With here the volume, the volume corrected by the intracranial volume, the signal to noise ratio and the contrast to noise ratio. For next, please, different drug effect and sample size. And here, for instance, if you're looking for a 30% effect of a drug for a sample size of 90% power, then to detect a change in the volume, we need about 120 subjects. It's much more with a signal intensity. And this was only a single-site study, and it needs to be reproduced on a multi-site setting. And next, please. Also, if we want to determine whether neuromelanin, dopamine, and iron are related in the substantia nigra, when we can have a look at the metabolic pathway of dopamine, where dopamine is oxidized using iron. It aggregates using different types of products, melanin, pheromelanin, and then it ends up into the neuromelanin organelles, which includes proteins, iron, lipids, and neuromelanin. So iron is linked to neuromelanin. And next, please. This is the case with imaging as well, where you have here this study from the group of Leipzig in Germany, where they have compared histological slices with MRI slices. And here on the upper image, what you see outlined in blue is the nigrosome 1, which is affected early in the disease. And you have an R2 star map, which is an index of the content of iron. And you clearly see that there is a lot of iron in nigrosome 1. They have extracted iron from this map, and on the lower image, you can see clearly that the yellow color has disappeared because you don't have iron anymore. And next, what they have done also is to compare the R2 star map on the left with the neuromelanin map on the right. The neuromelanin was obtained with optical density, and the R2 star map with iron imaging at 11.7 Tesla. And what you can observe is that the arrows are pointing exactly at the same place. So you clearly have, next please, a correlation between the location of iron here and the location of neuromelanin in the substantia nigra. So clearly, colocalization of iron in the substantia nigra with neuromelanin. And the next, please, what we have done at our group is to study quantitative susceptibility mapping correlation with neuromelanin imaging, both with an MRI. And here also you see a voxel-based comparison, a voxel-based correlation between iron content and neuromelanin in the substantia nigra. And the next, please. Well, the missing points for neuromelanin sensitive imaging is the fact that we have a lack of validation on large multi-center databases, calculation of sensitivity, specificity, time by group interaction, sample size needed to detect one-year changes. We need to better characterize changes in neuromelanin with the effect of disease duration, the severity of motor symptoms, the clinical expression with the different types of expression of the disease, age checks, and genetic status. We have seen the importance of the polygenetic risk score. We need also to determine the best acquisition and data analysis procedure. That will be the next talk. And we need to design and validate machine learning methods to predict the evolution of patients using baseline data. And I will end up with that slide, which is a summary. Next, please. Well, neuromelanin sensitive MRI measures neuromelanin content in dopaminergic neurons degeneration as well. It's a good cross-sectional marker of PD versus healthy volunteers at the group and individual level. It's relatively stable in controls around 50 to 60 years of age. It tracks disease progression. The changes occur at the prodromal stage. It's associated with clinical changes. The changes correlate with striatal dopaminergic dysfunction, and also they co-localize with increased iron changes. And I'd like to thank on the next slide my colleagues and you for your attention.
Thank you so much, Stéphane, for that really insightful presentation to the clinical relevance of neuromelanin in Parkinson's disease. Now, before we move on to Vahid's presentation, we actually have a poll for the audience. So if we can pull up that poll now. I think we can't quite see the question yet, Janie.
Yes, I do.
Yeah, it's all launched.
Okay, great. So the question is, what sequences do you know that are neuromelanin sensitive? Hint that it is a multi-choice. So I will give a few moments for everyone to take some time and think about what they think the answer is.
So far, all of the above is scoring at 65%, with multi-echo gradient recalled echo at 29% and magnetization transfer gradient recall echo at 30%. T1 weighted multi-slice turbo spin echo is at 11%.
I guess the answer will be in the next talk.
Yes, I think we can wait a couple of more minutes, maybe, or 30 more seconds, perhaps. I think it'll
I
be
think,
interesting for everyone when I reveal the answer.
I mean, it's peaked now. There's no more questions coming in, answers coming in. So I'm going to share
Perfect.
I'll share it now.
Okay, great. So it looks like all of the above was the most popular answer. The actual answer that's correct is options 2 and 3. So magnetization transfer gradient recalled echo and T1 weighted multi-slice turbo spin echo. With that, I will hand over to Vahid, who I think will prove very informative for you all, for those of you that didn't get the answer right. But over to you, Vahid.
Hi, can you hear me?
Yes, we can.
Cool. Okay, interesting result. Hi, thank you for the introduction, Hiba, and thanks to Stéphane for his wonderful talk. I'm Vahid Malekian, MR physicist and biomarker scientist at IXICO. As we saw in the previous talk, neuromelanin sensitive MR is an emerging technique that addresses the unmet needs for early imaging biomarker for Parkinson's, as well as tracking the disease progression. Now, in this part, I would like to bring your attention to neuromelanin MRI from technical perspective. Next, please. Here, I will discuss two key technical aspects of neuromelanin MRI that are essential to extract biomarkers. First, I will talk about the acquisition and second, analysis. In the acquisition part, I will explain how to image neuromelanin tissue, a dark pigment as seen in this cartoon, which is produced as a biomarker of the norepinephrine and dopamine metabolism, mostly in locus coeruleus and substantial nigra. I also discussed the key methodological considerations that need to be addressed for this type of acquisition. In the second part, which is analysis, I will walk through the analysis part, which includes tissue segmentation and extract the meaningful endpoints from the acquired image, which are ultimately used as a biomarker in the clinical studies. Next, please. Okay, let's start with the image acquisition. First, I would like to start with a review of major benefits of using MRI in brain applications. Most of you are already familiar with the fact that MRI is a non-invasive imaging tool that's widely available across the globe, making it ideal to use in large-scale longitudinal clinical trials. Another key advantage of using MRI is its multimodal capability. It can capture brain tissues with high spatial resolution based on their distinct physical and biological properties using different MR weightings, such as T1, T2, T2 star, proton density, and so on. But what happens if you use a standard structural MR sequence to capture neuromelanin-rich tissues such as substantial nigra? You will notice that substantial nigra is not visible in the midbrain or so-called Mickey Mouse region in the left figure. But we apply advanced MRI techniques that are neuromelanin sensitive, the substantial nigra becomes clearly visible as shown in the right figure. This highlights the importance of selecting the appropriate acquisition method to successfully detect neuromelanin tissue. Next, please. By this introduction, we now understand that the key objective here is to identify which type of MR acquisition are most sensitive to neuromelanin. However, before diving into the acquisition detail, it's important to first understand what's the main challenges associated with neuromelanin MRI. I'll start with the neuromelanin tissue structure and its associated MR contrast. There are several publications on this topic exploring contrast mechanism related to neuromelanin and also discussed in the previous talk by Stéphane. However, the exact mechanism remained a topic of debate due to the complexity of this structure. The second major issue is the challenging size of the neuromelanin structure, making them extremely difficult to resolve with the standard imaging protocols. The size is especially challenging when it comes to locus coeruleus, which you can see in this figure as the two hyperintense dots. Third important consideration is the location of these tissues. Both the substantial nigra and locus coeruleus are located deep in brain stem, as shown in the whole brain figure. Their anatomical positions present additional imaging challenges such as sensitivity to flow and motion due to their proximity to a force ventricle and large blood vessels in this region. So, based on the challenges mentioned, I will present potential solutions starting with a brief explanation about the neuromelanin contrast in MRI and its associated pulse sequences, followed by strategies to optimize imaging protocol to achieve high resolution, high quality images. Next, please. Starting with the contrast. We saw earlier an example which substantial neurogrowth was only visible in one of two MR images, which we refer to as a neuromelanin sensitive scan. This raises an important question. What is neuromelanin sensitive scan? To address this, we need to look at the literature. Recent quantitative MRI studies mostly report two main tissue property differences between neuromelanin-rich and neuromelanin-free regions, which can be captured by MRI. The first difference is a macromolecular to water content ratio, which appears lower in neuromelanin-rich regions compared to its surrounding. In the left panel, by looking at the pool size ratio or PSR map, which quantifies the ratio of the macromolecular to free water pool, shows that substantial negra has a lower PSR value than its background. This difference can be detected via magnetization transfer process or MT in MRI. The second difference lies in the neuromelanin iron complex structure, which affects T1 relaxation time in MRI. As shown in the T1 map at the right panel, the substantial negra exhibits shorter T1 relaxation time against its background. By looking at the bottom cartoon, you can observe shorter T1 means higher signal or brighter intensities in the image. Next, please. So, based on the MR ratings that are sensitive to neuromelanin-rich regions, two commonly used sequences are MT-based gradient recall echo, or MTGRE in brief, and turbo spin echo, or TSE in brief, which is the answer to the poll. MTGRE is the standard gradient echo sequence with an added MT pulse at the beginning, as shown in the MTGRE sequence schematic. MT pulse selectively saturate macromolecular contents due to its difference in resonance frequencies from free water pool as indicated in cartoon in the center of the slide. This effect is clearly demonstrated in the bottom figure when substantial negra is visible only in MTGRE, but not in conventional GRE image, highlighting the importance role of explicit MT pulse in MTGRE sequence. The second candidate is a T1 weighted turbo spin echo, or TSE. This TSE based sequence also benefits from incidental MT effect, which generates by refocusing pulse in the sequence, which also shown in TSE sequence schematic. So, both T1 and MT effect may contribute to the neuromelanin contrast in this sequence. To briefly compare these two sequences, TS is easier to implement while MTGRE needs more parameters to consider for protocol harmonization due to the variability of MT pulse settings across MR vendors, such as off resonance frequency, amplitude, duration, and shape. However, TS is slower and more SAR intensive, especially when explicit MT pulse will added into the sequence, which is called MTTSE. And SAR stands for a specific absorption rate, refers to the rate at which energy is absorbed by tissue. High level of SAR can lead to the potential scan limitations. So in summary, it's important to carefully select the acquisition method based on the study's requirement and specific research question. Meanwhile, it's worth noting that here, 2D MTGRE was the sequence used in PPMI project. Next, please. Okay, now we understand NM sensitive sequences. The last step is to find out how to properly set up the optimal protocol. At the center of this slide, you can see that the well-known trade-off triangle in MR physics. highlighting the balance between the signal-to-noise ratio, spatial resolution, and acquisition time. Optimizing the neuromelanin MRI protocol requires careful consideration of a trade-off between these parameters. As we saw, high-resolution imaging is essential in neuromelanin MRI since both substantial nigra and leukoceros are tiny structures. However, achieving high resolution comes at the cost of longer acquisition time and lower SNR. As you can see, the impact of lower SNR images on substantial nigra visibility in the top figure. This would be even more challenging for Lucus ceruleus, which is a smaller and more difficult structure to visualize. So, to maintain high resolution while ensuring acceptable SNR, it's often necessary to repeat the scans and average them to reduce thermal noise. For example, in PPMI project, neuromelanin MRI scans were repeated 5 times to boost the SNR. However, there is no such a thing as a free lunch. The downside is to longer the acquisition time, which increases the patient burden and risk of artifacts such as head motion. So the key takeaway here is that the need to balance these competing factors to achieving high image quality within reasonable scan time. Next, please. The trade-off triangle is not the only thing to consider. More sequence parameters are joining the party. For example, if you are scanning both substantial nigra and Lucus ceruleus at the same time, optimizing sequence flip angle is something that needs to be carefully considered. Substantial nigra and Lucus ceruleus have different optimal flip angles to visualize, as seen in the red boxes in the left figure. This is because Lucus ceruleus, also located near the CSF or fourth ventricle, require contrast not only against its surrounding, which is pontine tegmentum, but also against CSF. As an example, I've included a figure from the previous research that we conducted to improve Lucus ceruleus visualization at 7 Tesla, showing how adjusting the flip angle affects Lucus ceruleus visibility and its associated contrasts. Next, please. The final point in the acquisition part that I want to mention is the field of view planning. Aligning the imaging plane perpendicular to the floor of fourth ventricle minimizes partial volume effect, while aligning the plane with ACPC line, which is more familiar to people, may be more practical, easier, and offer better reproducibility for multi-site studies. The other thing is the limited brain coverage. And why we do the limited brain coverage here? Because to save time to make the scans shorter. But limited brain coverage is another factor that carries a risk of accidental exclusion of the part of the neuromelanin region. So a clear, step-by-step operational imaging guideline for MR technologists is essential to ensure consistent field of view placement across MR sites. Next, please. Now we are moving into the second part, which focuses on how to analyze neuromelanin MRI images after acquiring them. Analysis typically involves 3 main steps. First step is the pre-processing, which commonly include motion correction, brain extraction, denoising, and averaging the scans to make the high-quality final image. The neuromelanin image before and after processing shown in the top and middle images that you can see the effect of pre-processing on substantial nigra. Second step is the segmentation, to segment the neuromelanin tissue. The result of this step can be seen in the bottom figure. This step can be performed using various methods such as manual, semi-manual, template-based, or deep learning approaches. I will not go through the manual methods because of their scalability limitation, but I will talk about the template-based and deep learning approaches in brief in upcoming slide. The final step is the quantification, which can be divided into categories: volume-based and contrast-based metrics or intensity-based metrics. In this section, my focus will be on substantial nigra segmentation as a main neuromelanin region in clinical trials. Next, please. The first common approach to segment neuromelanin tissue is a template-based method requiring two-level registration, from native neuromelanin MRI space to the patient's T1 imaging and then to the atlas space, as shown in the left figure. The substantial nonogram mask from the atlas is then transformed back into the native space to segment the tissue. This process may need further post-processing to improve segmentation accuracy. The second approach involves AI-based methods such as deep learning, which requires training dataset to learn the features necessary for tissue segmentation. One example is NagraNet, shown on the right figure, which is developed in Stéphane's lab to segment substantial Nagra. Now it's worth noting some of the key pros and cons of each method. For template-based segmentation, it's relatively easy to implement with a lower computational cost. It doesn't require training dataset and generalizes well across neuromelanin acquisition methods. However, it relies heavily on the accurate registration, availability of the T1 weighted data, and atlas images that may need also some post-processing to refine the segmentation results. In contrary, AI-based approach is fully automatic and potentially more accurate analysis method without needing T1 image or atlas images. However, it requires a training dataset and its performance depends on the quality of the training and model selection. Next, please. After segmentation, it's time for quantification. A volume-based measure metrics such as area, volume, or width of the neuromelanin region are mostly reported aligned with the normalized volume with respect to the head size or total gray matter. In the contrast-based measures, relative signal-to-noise ratio or contrast-to-noise ratio are mostly calculated and reported. In this category, we need to use reference region to calculate the baseline intensity. For substantial Nagra, the reference region is typically cerebricus, while for lucus ceruleus is pontine tegmentum, commonly used for reference. As an example of using these metrics in PD studies, Martinez and colleagues show significant substantial Nagra volume loss and contrast reduction in PD cohort compared to the controls. We also saw lots of examples in the previous talk regarding these biomarker results. It's also worth noting here that the recent findings in the PD clinical trial have shown exciting evidence that the neuromelanin biomarkers are sensitive to detect trends toward delaying motor progression, highlighting its high potential in MRI-based PD clinical trials. Next, please. All right. Now it's time to wrap up the talk by summarizing a few key points. First, MRI is a non-invasive and powerful tool to capture neuromelanin. T1 and MT-weighted base sequences are mostly commonly used in these studies. Lucus ceruleus imaging needs more consideration in protocol setup. Harmonized protocol and field of view positioning are crucial to acquire high-quality images in large-scale studies. Volumetric and contrast-based measures are the primary neuromelanin biomarkers. And finally, pipelines using AI boost the power of neuromelanin MRI data analysis. Next, please. Okay, thank you very much. Back to you, Hiba.
Thank you so much, Vahid, for that really detailed presentation from a technical perspective. I believe we can actually now move on to the Q&A session. We have a nice amount of time for it, and we also have a nice amount of questions. So please allow me to welcome back Stéphane as well. Welcome back, Stéphane. And we can start answering our questions, I believe. So we've got one here, Stéphane, for you, which relates really to the pathophysiological evidence. And the question is there pathophysiological evidence that the increased MRI melanin signal in PD or models is sufficiently reduced therapeutic candidates to warrant surrogate endpoint nomination?
Well, it's an important question. We have a number of good points for neuromelanin imaging. And among these good points, we have the fact that it is an index of catecholaminergic neuron degeneration. It has been shown histologically. It's early, it's longitudinal, It correlates with clinical symptoms. It's relatively stable in controls. And we need, on that one single site study, moderate number of subjects to detect changes over 1 year. But we still have work to do. For instance, what sequence is best, as shown by Vahid. We need to investigate the multi-site effects, I think, which means that we need to have this power calculation on a multi-site setting. It's also difficult to read in clinical practice without a software, which means that we need to have a software to do automated calculation of the volumes and signal intensity. And the mechanism of contrast is still debated. But I guess this one is probably not an issue, because even if the mechanism of contrast is debated, then if it works, we know that it's a good surrogate of catecholaminergic neuron degeneration. So in my opinion, if we have a drug that targets to reduce neurodegeneration of dopaminergic cells in the substantia nigra, then I guess neuromelanin is a good candidate, even though we need to investigate a bit more on a multi-site setting.
And I think actually, and there's quite a few questions relating to this, and I think even our next one is slightly related. And the question is, do the panel think there will be a means to standardize neuromelanin imaging acquisition and analysis across centers in the near future to enable global studies to incorporate this methodology? I suppose there's really two parts in terms of what can be done, and secondly, is it practical? Can we actually implement what needs to be done on a global scale? And I wondered what your thoughts were, both Stéphane and Vahid, on this.
This has been done already, because if you want to conduct a multi-site study with neuromelanin imaging, what you can do is to optimize for each one of the scanners of each one of the vendors the sequence parameters, so to get images with a good contrast to noise ratio for substantia nigra. This has been done in several studies, at least that I know, in France. And I guess also IXICO perhaps, but Vahid can better answer that question.
Yeah, thank you. This is a big question, and Stéphane addressed it quite well. The key point that I can mention is like a standardization of the sequences and the parameters that Stéphane mentioned is already done in a few studies. So yeah, doing standardization in the hardware, in the acquisition, pulse sequences, parameters that I mentioned during the talk, and also the analysis pipeline is quite important. And at the end, like the way of quantifications also need to be considered to do the standardized or harmonized studies.
And then you have a lot of post-processing methods. You can use combat. You can use many types of different statistics to try to also overcome the differences of the images between the different vendors.
That's true. Yep.
Right. And our next question we have is for Vahid, which is again relating to acquisition. So what is the typical acquisition time for neuromelanin MRI? And what are the options to make acquisition as fast as possible to reduce patient burden?
Okay. Yeah, that's a great question. I think it's like I can just make an example of the PPMI project. It takes, I said it's a 5 repeat. It needs 5 repetition, and the total time would be about 10 minutes. But it really depends on the protocol that you're gonna set up. It depends on the spatial resolution. It depends on number of repeats. It depends on the coverage. It depends on the slice thickness. So it really depends. The acquisition time really depends on the parameters that you're gonna set up into the sequence, as well as the sequence itself. So I would say, like, just as an example for a rough idea, it would be you can just push it to 6 minutes up to around 12-13 minutes.
It's also our experience. We started in research with an acquisition about 7-8 minutes perhaps. And now in the clinic, we also use neuromelanin imaging with deep learning denoising, because the vendors now offer that type of denoising, and it's down to 5 minutes. So it's really a clinical sequence.
And I suppose from a patient burden point of view, that's not very long at all as well. So I suppose if it's definitely used on its own, it's fine. Even alongside other sequences, it wouldn't pose such a problem in terms of timing and patient burden.
Right, you can certainly use a sequence of 5 minutes duration acquisition time in a patient. And what we do is also to combine that with a 3-dimension EPI to get the swallowtail sign.
Yes.
That's a susceptibility weighted image or strongly T2 weighted image, which is with a high resolution. It's a 0.5 millimeter isovoxel size, and with that you can get also the swallowtail sign. And if you combine neuromelanin and swallowtail, it's very useful comparison to that scan, for instance.
Yeah, that's a great point. But the thing is, like when we're using the very advanced type of acceleration techniques, the risk is a little bit in harmonization. So it might be like some of the sites it's not available in some site, but not in others. So for large scale multi-site studies, it might be an issue of a harmonization.
Yes, I was just thinking about our experience using a recent scanner.
Stéphane, you mentioned the swallowtail sign, and we actually just had a question come in. From a clinical neuroradiological point of view, is there still room for the swallowtail sign? Do you use it in clinical practice? How about sensitivity and specificity?
Very similar between both, but it's easier to read the swallowtail sign on susceptibility weighted image or 3D EPI than to read the neuromelanin image. Because to read the neuromelanin image, you need to have seen a lot of control subjects and to train yourself to read that type of image. For the swallowtail sign, it's present or absent. It's easier to read. But now we have also developed an automated method to determine whether we have the swallowtail sign or not. And then if we combine, we haven't the results now, but if you combine the nigranet for the neuromelanin and the nigronet for nigrosome 1, which is very similar to the swallowtail sign, then I guess you're more than 90% accurate to determine whether you have neurodegeneration in the nigra. But this is a sign of neurodegeneration in the nigra. It's not a sign of PD, because you find the same type of changes in a typical Parkinsonism. So you cannot use that to distinguish a typical Parkinson from PD. But to distinguish, for instance, a non-degenerative Parkinsonism from degenerative Parkinsonism.
I suppose it's a little bit like the DAT-SPECT, where you can't use it to differentiate between the atypical syndromes as well. Perfect. Vahid, we have a bit of a big question here, so without going into details, perhaps you can give an overview of what are the MRI scanner requirements for neuromelanin imaging in terms of sites and scanners, which sites and scanners can perform neuromelanin imaging?
That's a good question. Yeah, I mean, mostly focus on the 3 Tesla scanners to get a good type of signal-to-noise ratio with the acceptable spatial resolution and within the reasonable scan time. As I previously mentioned, there's a famous trade-off triangle in MR physics, and we know that we need a spatial resolution. These structures are tiny, so we need to just in terms of the scanner requirement, we need a kind of a good hardware, and starting with the 3 Tesla would be a kind of a standard field to do the neuromelanin imaging.
So at a bare minimum level, having the 3 Tesla scanner.
That's correct. Yep.
We have, I think, Vahid, this might be one for you as well. In the voxel-based intensity analysis, what spatial normalization approach do you use? And is there any intensity normalization or standardization needed?
Well, go ahead, Vahid.
Yeah, you go ahead, Stéphane.
Yes, well, you can use any type of co-registration that works and normalization. For instance, ANTS would work, or SPM, or whatever software that provides good results. And for the intensity correction, yes, of course, that can be used.
Yeah, exactly. So it's like mostly for neuromelanin MRI space to T1, if you want to use a template-based co-registration and you want to do the co-registration between these two space, we mostly use like rigid type of a registration with a 6 degree of freedom, and from from T1 weighted space to the atlas space, we mostly use like a non-linear type of a registration to do a kind of a good registration, and then concatenate these two transformation together to make a final transformation from atlas to the neuromelanin space for the accurate co-registration. And also for bias correction, we use different techniques to remove any residuals related to the intensity and homogeneity in neuromelanin images.
Perfect. I can see we've got a few minutes to go, and with that, Stéphane, I have a big question for you from the audience. Where is the future of PD clinical trials going?
We have we have some in France going, and what we are doing now is to try to get all the neuromelanin images of many different trials. These are not all drug trials. These are sometimes mostly pathophysiological trials, and so far what we have obtained is about 1,000 scans with a turbo spin echo sequence. Not all of them have been collected so far, but this is a multi-site setting, at least 6, 7 centers, and 1,000 is a lot, which means that we'll get a better evaluation of the power calculation and the changes of disease duration, types of Parkinson's disease from severe to mild, and so on. So it's a study which is ongoing.
Wonderful. Thank you so much for that. We've got lots of questions coming in. I'm sorry, we will have to answer some of these over email, but thank you so much for everyone's questions. I want to thank everyone so much for their participation. We hope you found today's webinar insightful. Please do take a brief moment to fill out our survey that we'll have at the end. If you missed any parts of the webinar, don't worry, we will have this recording available for you later on. And with that, thank you so much for joining, and thank you to our wonderful speakers, Stéphane and Vahid.
Thank you very much.